UC San Diego Stem Cell Program and Scientific Director of our home that you find yourselves in tonight, the Sanford Consortium for Regenerative Medicine. This building, for those of you who don't know, is a very unusual and wonderful experiment in the development of San Diego's tech history, I hope. It's a collaboration between 5 separate academic institutions on the La Jolla Mesa, UC San Diego, the Salk Institute, the Sanford Burnham Medical Research Institute, Scripps Research Institute, La Jolla Allergy and Immunology. And we've also are indebted to Denny Sanford, who made a very generous gift to help get this built, along with a very generous grant from the California Institute for Regenerative Medicine. The chairman of the board, John Thomas, is here joining us tonight. And if John's lucky, he'll make it out of here without my doing one of my customary experiments on him. But we'll see. I reserve the right. Those of you who have not been part of this 3-day festschrift of stem cells, medicine, business, and technology, I'll just tell you that every year for the past, are we up about 10 years now? 7, okay, getting towards 10. Last 7 years, we have had what started as Stem Cells on the Mesa, or Stem Cell Meeting on the Mesa, SCMOM, for short, for those of us in the know. It's now evolved not only to a scientific meeting, but also to a business and partnering meeting as we discuss and in many cases move stem cell-derived treatments towards clinical application. And this year it is a full 3-day affair. We are 2/3 of the way through. Tomorrow will be the scientific meeting part of it, and the past 2 days have been the business part. For those of you who are not attending the scientific part tomorrow, I'll just tell you that you're going to be missing something truly fantastic, because there will be talks about the role of transformation of cancers into stem cells by genetic mutation to drive cancers. There will be talks about how we're currently using stem cells to try to understand disease and develop better drugs. There will be talks about how to use stem cells directly as therapies, very early stage, as well as a very rapidly developing new area, some of which is happening in this building as well as the others, which is the development of novel biomaterials to coax stem cells into the formation of ultimately artificial organs, we hope, built from cellular materials. I sometimes tell community groups that they have arrived in the plastic factory, where stem cells are the biological plastics of the 21st century. Yes, Frank, I got that right. And that in the coming decades, you'll see biological materials be very much like plastics were for the past 50 years, where you can build almost anything of biological use from these materials. Now, for those of you who couldn't squeeze into this room, and I presume you're not in this room with me now, but for those of you who get tired of standing up, we are broadcasting downstairs in the lobby, out on the veranda out here, which is where the reception will be, and you are welcome to stay here or move there if you would prefer to be more comfortable. So that's all I want to do in the way of general comments and welcome. I now want to turn to one of the more amusing parts of my day today, which is to introduce Craig Venter. So this now punctuates our 3-day festivity at the 2-day point. In many ways, introducing Craig is one of the more unusual experiences I've ever had, since I would count Craig as friend, collaborator, colleague, and experimental subject. So those of you who visited my lab in the past hour saw us growing Craig's brain cells in a dish, ready for transplant to any worthy recipient who can tolerate the many strange disturbances in the force that we're discovering in these cells. And I think Craig will have more to say about that. Let me give you some of the facts and figures on Craig. I mean, he's a legendary scientist of our era. He's currently founder, chairman, and CEO of the J. Craig Venter Institute, which is based partly in Maryland. I don't think Maryland exists anymore, last time I checked CNN. So that's good news for us, because he's moved part of the institute to San Diego, and I'm hoping that the storm will persuade him to move the rest of his institute out to San Diego, because they've been very dedicated to understanding genomes in both humans, model organisms, and of course plants. He's also CEO and founder of Synthetic Genomics, which is a local company that's trying to effectively build artificial organisms using artificial genomes to try to develop novel foods, novel sources of energy, and presumably lots of interesting things that none of us have thought of yet that might be done with these kinds of unique materials. Craig's history is unusual. He was a Navy corpsman in Vietnam. He was then an undergraduate student here at UC San Diego. He left a few years before I got here, or I should say, he got out of here just before I arrived. After earning his bachelor degree here, he also received his PhD here with Nate Kaplan, and then went to the State University of New York at Buffalo, where he worked on receptors of various sorts. He then moved to the NIH, where he really began to pioneer what I think most of us would recognize as the modern era of genomics, with the realization that you could use so-called expressed sequence tags to very quickly find genes, enumerate genes, and look at how cells were behaving using very rapid sequencing technologies. And this ultimately led to his founding Celera Genomics, which completed the genome sequences of a number of model organisms, including one of my favorites, the fruit fly, ultimately the human, my second favorite organism, and a lot of other bacteria, fungi, and strange marine creatures along the way. Blah, blah, blah. Okay, so, it's not just me who thinks he's really terrific. He's won a number of really amazing awards. The U.S. National Medal of Science, the equivalent of the American Nobels, the Gardner Foundation Award, and he's a member of a number of prestigious academic societies. Now, one thing I like to do when I introduce friends and colleagues is I like to go through their CVs and find things that I regard, not them, but I regard as interesting. So in his 50-page CV, I found that, let's see, on page 16, one of his most important papers came out in the year 2000 called Comparative Genomics of the Eukaryotes. That paper is important because it's the only paper that Craig and I are co-authors on. I'm buried somewhere in the middle of 30 authors. Craig's right down there near the end, which signifies that he actually did something important. The year 2001, sequencing of the human genome, that was an important one, you gotta admit. That got a lot of fanfare. The complete sequence of the human genome, which he will tell you some about tonight, is an incredibly important accomplishment. And then a paper by Levy et al. In 2007 is ultimately the reason why one of my students followed Craig on my student's motorcycle. I think you were driving a Maserati at the time, and persuaded Craig to let us biopsy him. And so, because of the existence of his complete DNA sequence, we reprogrammed him so that we can have his brain cells in a dish in the lab to study and do various interesting things too. We've been insulting him in various ways. And then more recently, the report that you can chemically synthesize a genome of a bacterium and get the bacterium to actually run off of that chemically synthesized genome. Interesting books and publications about Craig. Another one